Okay, so this is chapter 16, part 1. And with evolution, you guys, people get really confused of what it is. And I just want you to really know this definition inside and out so that you are not confused anymore. Jake, go for me. So evolution is just the change in populations through time. All of us are very comfortable, I think, realizing that organisms change over time, okay? Now, there are two main types, okay? And that has to do with time. And micro is what? Small. And macro is large. And we, are on Earth a very short amount of time. So micro, we can see. It's pretty awesome. Like, you guys know of things like AIDS evolving, the flu virus evolving, bacteria evolving. We can see things change. That is actually called microevolution. It's also known as population genetics. Did I make you guys write microevolution right up here big? Okay, so chapter 16 is all about the evolution that we can see with our eyes, we can measure. Okay, now macro, we can see the products of it. So the video with the finches, I think 13 different species of finches on the different islands of the Galapagos archipelago, the chain of islands there, the group of islands. So were we able to document and watch the ancestor of all 13 of those species? We couldn't see it happen, right? No, but we can look for clues and we can find evidence to study speciation, which is macroevolution. Okay? So I want to first just concentrate on micro with this chapter. The other chapters will talk about macro. And actually, the video talked about micro, what the couple saw, those two scientists, and they also talked about macro, especially that last 5 minutes with what I just talked about with the 13 different species. Okay, this is hard to see. I made it small because I was just trying to abbreviate things for you guys. But listen carefully, and you'll know what he's putting in the little swimming pool. And then I will also explain it.
Of all of the genes and their different alleles that are in a population. The gene pool is a collection of all of the genes and their different alleles that are in a population. In simple population genetics models, the gene pool is imagined to be an infinitely large body of water containing all the alleles. For example, if a population of plants consists entirely of homozygous purple-flowered plants, then its gene pool has many copies of alleles that make purple flowers, and no alleles that make white flowers. In this case, the frequency of the allele for purple flowers is 100%, and the frequency of the allele that makes white flowers is 0%. If the population consists entirely of homozygous white-flowered plants, then the frequencies of alleles in the gene pool are reversed.
Okay, so big P's. If there's 100% homozygous big P's, okay, then the gene pool only has big P's. You guys got to get this concept down right now. So it's not a scary thing. There's only big P's or little P's. Okay? So then he switched and he said, no, there's no variation. They're all little P's. Now what about heterozygotes? What if they were 100% heterozygotes? What would the gene pool be made up of? 50% what? Big P or small p, 50% the other. Okay? So this is very simple math. We're going to be doing some math during this chapter.
100% alleles for white flowers and 0% alleles for purple flowers. It is also possible that both types of alleles are present in one gene pool.
Okay, so we talk about these in percentages or we talk about them in decimal form. So they add up to 100% if we're talking about percentages. If we're talking about them in decimal point, it adds up to what? 1, right? 0.5, 50%, right? Plus 0.5, 1, right? Okay? So gene pool is a very important concept for you guys to get. It's just all the alleles in whatever population we're studying. So with the finches, there were some alleles that made, some genes that was an allele that made for making a larger beak versus a smaller beak. I'm sure it was not as simple as purple flowers and white flowers. Maybe more like our chin stuff too, where there are multiple genes that are part of that. I'm unsure of that particular trait. But a beak is a pretty important organ, and it's a lot more complex than this color or that color. You guys following me? Okay, so the gene pool, did it change for the finches? Yeah. Okay, so here's a nice example. Back to the peas. Listen to this one.
Homozygous purple-flowered plants are crossed with homozygous white-flowered plants to produce heterozygotes in the F1 generation. Note that the frequency of each allele is the same, 50%, in the F1 generation as it is in the P generation. When the heterozygotes in the F1 generation are crossed, the frequency of each allele remains the same.
Okay, so this is very important for you to note. So we started with half and half. Let's say half of them were big P, and half of them were little p, little p. So then what's the allele frequency for the P, big P? If they're half and half, the big P is 0.5 or 50%. Now, I brought this up on purpose. Some of you might have went to the genotypic. Right now, hear me. When you're doing these problems, you have to know, are we talking about the allele frequency, the number of big P's, or are we talking about the genotypic frequencies? Because there's 3 different genotypics possibilities. Homo, hetero, homo. So there's 3 options if we're talking about the amount of genotypic frequencies, which makes the phenotype. But we could also just talk about the P's by themselves. That's the allele frequency, and it's either a big P or a little p. So we only have 2 options. Okay, so please make sure you know what the questions are asking you. Now, has the allelic frequency changed? Has the number of big P's changed in this example? No, if you count them, right? So we're going 50-50. Now we started out with 2 parents, right? But look right here, big P, 1, big P, 2, 3, 4, 5, 6, 7, 8. How many little P's are there? 8. Still 0.5 and 0.5. There has been no evolution of the alleles. Now, yes, the frequency, because of dominance, did change. There's more purple than white, okay? Okay, so this is just a vocab word here on the top, polymorphic. It's just saying that there are many, or at least more than one, different type of morph or trait or that organism. Like that one had purple flowers or it had white flowers. So none of this stuff works very well if we can't see it, if there's not these different morphs. We wouldn't be studying those traits. And maybe we don't have to see it with our eyes. Maybe we can detect it, though, other ways. Now, the 4th bullet, star it, underline, highlight it. This is hugely important. Okay, this is one of the biggest take-home things that you should know about biology, specifically with evolution. Okay, so the ultimate source of variation is what? Mutations. Evolution gets rid of variation most of the time, because if the environment, if it's better for an organism, say, to be white, maybe it's camouflaged or something, I don't know, so then the deer can't see it and find it and eat it, I don't know, okay? So hold on. So the deal is a lot of times natural selection is getting rid of variation. We don't want the sickle cell allele, for example. Well, people that get the sickle cell allele, get 2 of them, they die, so we're getting rid of the variation. The only way to make variation is 2 different ways. Again, what's the first one? Mutation. The second one has to do with sexual reproduction. So it's the recombination of alleles during meiosis. What event am I talking about? Crossing over and the segregation of the homologs. Dads versus moms. And we're doing that every time for your 23 pairs, so you're not going to look just like your sister or brother, right? This is making variation. So it's the shuffling of genes, the book calls it in this chapter. But you need to be able to identify it through meiosis making variation, through segregation of homologs into the gametes, or the crossing over, which all has to do with sexual reproduction. Okay? Yes?
So these are sexual is just get the withdrawals, kind of like
Okay? So genetic variation is very important to the health of a population. Okay, so let's look, this one's a nice little review.
The genomes from two different individuals are 99.9% the same. This means that the 3 billion base pair human genome is expected to contain about 3 million variable sites. One source of all that variation is mutation. Mutation gives rise to new alleles, such as when capital A mutates to lowercase a. Before mutation, only capital A alleles were present. The second source of variation is the recombination of alleles during meiosis. Crossing over generates new combinations of alleles at different genes. Before meiosis, only the combinations capital A lowercase b and lowercase a capital B occur. Crossing over between the two genes results in the recombinant types capital A capital B and lowercase a lowercase b.
Okay, so now, these two individuals, Hardy and Weinberg, figured out some math that could help us measure the amount of evolution that is occurring in different populations. So, it's a very famous equation. Now, for it to work, we need to get to a baseline or to a zero. So you need to remember the assumptions of Hardy-Weinberg's model. Now look up here. So if we're going to use Hardy-Weinberg, it needs to be in this situation. The first one, we're talking about diploid organisms, right? They have to have an option of a genotype, 2 alleles, big A, right, for example. Okay, the other thing is generations are not overlapping. So that means parents are not mating with the second generation. Now that sounds goofy and weird for us to think about things like this. It does happen, though, in humans. But also, for us controlling this, we need for the experiment, we can't have overlapping of generations. Now, it sounds weird for humans, but it's not very weird with plants and for insects. For example, if we're raising fruit flies in a test tube vial, and I stick some males and females in there, that's your parent generation. They make babies in a couple weeks. I mean, they're completely flying around. Now they're mating. You telling me the parent's not going to mate with an offspring in the fruit fly vial? They're going to mate. They want to pass on their genes. Okay? So, for Hardy-Weinberg to work, we cannot overlap generations. So we have to get rid of the parents before we let the breeding continue. We need large populations. Typically, for the same reason we want to do science with large sample sizes, right? It gets rid of the random luck. If you have a small population, randomly you could have ended up with more blonde-haired individuals versus brown-haired if you had a very small population, compared to if you had a big population. Okay, mutation is negligible. Now you might not like that word, so just put no. No mutations. We can't have any new alleles popping up to change the frequencies. So no mutation, nothing new. We're trying to make a zero. We're trying to make a baseline. That's why we're doing all these assumptions. Okay, reproduction is sexual. Okay, so we're not having any asexual. We need this mixing up of the alleles. Gametes unite at random. Now this also means just random breeding, random selection. That means there's no, like, the person with this trait is more appealing, that person's going to mate more. Again, that's not going to happen. Well, it can happen actually in the fly world. There are courtship behaviors actually with flies. But we're going to get rid of that. So no individual, no allele is better than another allele. Randomly mating. Okay? Then migration is negligible. We can't have any other flies coming in that are carrying different alleles than our population, because is that going to change our population? Yes. Okay, natural selection does not operate. One is not more fit than the other. This trait is not a better trait. Now, does this sound like this happens in reality anywhere? No. It's a baseline. Now, sometimes when people read this for the first time, they're like, well, this is a piece of junk. What do we use this for? As a baseline. So we're looking at a 0. No evolution is going to change. The population is going to stay the same with these assumptions. So that's how this is useful. Okay? Now, here's the first part with the math. And don't be scared if you're like me. Math can be scary. I'm going to start nice and fresh over here. Okay, so the main equation is P plus Q equals 1. Because there's only P and Q. Now, what is P and Q? So P plus Q equals 1. P stands for the frequency of one of the alleles. Let's go with the dominant one. So you guys want to go with the big P? Yeah. Okay. And this one's the frequency of the little p. So they simply said P will always stand for the capital letter, Q will always stand for the, which is the recessive allele. Okay? Now, write down this somewhere because it's not written down. The whole equation needs to have 3 parts because genotypes, there's 3, right? Because of the diploid situation. So basically we just have to factor it. P plus Q factored with P plus Q. Now I'm showing you some math, showing off my math skills. You don't have to understand this, but I think it's a good little review for some of you guys. So what do I write there? That's a huge plus, huh? Plus what? I got my PQs and my QPs. Q squared. Pull these guys together, what is it? So Hardy-Weinberg's equation for the 3 possible genotypes are P squared plus 2PQ plus Q squared equals 1. That is your equation. Jot it down. Okay, now, here we're looking at alleles because we're looking at the whole gene pool. How many big P's are there compared to how many little p's? That's what frequency means. Okay? There's a brewer's blackbird on the feeder. I've never seen that happen. Do you guys see how big he is in the middle? Okay, now, for here, we're not talking about the individual alleles, we're talking about the genotypes. So P squared stands for the frequency of what? Big P, big P. Now you guys, this is hugely important. Listen to me. You're not going to have to use the quadratic formula. You're not going to have to unfactor this. You're going to use this thing in pieces. Very easy pieces. So that's why I want to make sure you understand what's going on here. Jacob, you with me? You can put that away. So this is big P's frequency is P squared. Big P, what is this representing? This is the heterozygous frequencies, 2 times P times Q. What is Q squared? Okay, now, most of the time, hear me, okay, we deal with Q squared. Why? Can we see Q squared with our eyes? If this is a white flower, we know its genotype, right? If it's a purple flower, it's both of these and we don't know, which it actually would be this total thing. I don't want to work with that. I just want to work with one piece at a time. Okay? So what Hardy-Weinberg is doing is it's allowing us to mathematically figure out the frequency of the two or the two other genotypes, or if you have Q, you can figure out what P is. So I've said all that. I'll talk about the pepper moss later.